Hey, hi there, Woolly and Pragmatic Crystal, and Scott Duke and Andrew. Woolly and Pragmatic Crystal, we pay them to show up.
Yeah, they're our filler audience.
Yeah, we pay them.
Loyal
It's like
followers.
priming the pump. You got to put a little water in the pump to get the water going.
Yep. They're actually actors, and they work as extras on a lot of movies, and I hired them to show up on my streams. I wish I could hire more. I wish I could hire like 50 of them, but they're so goddamn expensive.
Yeah, that costs a lot of
money. Okay, we got a little clip here from Patricia Engler. Where did she go wrong in life?
I think she just grew up in a lower middle class to upper middle class Christian Kentucky family, and just got stuck into that kind of belief, and has just never gotten out.
Yeah.
Ooh, there's a patriciaengler.com, 5 consequences of societies that forget God. Lessons from the Lantern.
Ooh.
Okay, what does lessons from the Lantern dude mean? What
Is it Green Lantern? Yeah, I love that comic book. I used to when I was a kid, anyway.
That's what it said in the YouTube thing. Teaching children that scriptures are true is nothing less than child abuse. No real scientist questions evolution or human origins. These are things that she's putting in quotes. She don't like that stuff.
Those are things she's going to overcome, huh?
There's too much hypocrisy in the church for Christianity to be worth believing. Religion is humanity.
Oh, she's overcoming reasons to not believe, so that you'll feel so ashamed of yourself for all your excuses that you fall upon your face and you cry out unto the Lord. Oh, I'm sorry.
Oh, you are a good preacher. You missed your calling.
Yeah, I did, didn't I?
Oh, let's see. Oh my goodness. So anyway, she's got that.
Oh, I was going to say after we. Sorry, go ahead.
I kind of shook you to your core there, didn't I, Andrew? Just kind of rattled you. Is it the Holy Spirit? Is it the Holy Spirit convicting you,
maybe.
Drawing you in?
I guess so. Yeah. Feeling that
preparing the ground for the seed of God, which is the gospel of Jesus Christ.
Oh wow, you would make a good preacher. Sorry. Go ahead, Speed.
She's a former homeschooler and BSc grad, Bachelor of Science C. Wait, what does a C stand for? That's just science, right?
I don't.
BFC grad, isn't that an institution then? If she's a grad from there.
I don't know. She has a threefold passion for Christian apologetics, outdoor adventure, and Dutch licorice. When I'm not writing about worldview topics, traveling out of a backpack and trying to build camping gear from dental floss, you can probably find me outside tuning a ukulele and eating licorice. Okay.
She has an eating disorder. Is that that's what we're discovering?
I think so. Yeah.
Okay. So let me see. Let's go to Google, and then
the credentials.
What college is BFC?
Oh no,
Bethany First Church? No.
No, I think it's Bachelor of Science grad. Bachelor of Science.
Oh, you said BS. I thought you were saying BF.
No,
as in Frank.
Yeah. BSc, Bachelor of Science, I think. Okay, you want to play the clip here, and let's get mad.
Sure. Yeah. Yeah, I want to review this video real quick and hopefully spend some time. I put together a little GULO with some examples of other pseudogenes that I find interesting for the whole evolution creation debate. So hopefully I can interesting little presentation there
Good.
for what together. Yeah,
we'll do
let's
this
go
and
over
then
this
we'll
video
do that.
first.
Okay.
Welcome to Critical Thinking Scan, where we look at how you can think about any phase-challenging message and arrive at a biblical, logical conclusion yourself. I'm Patricia Engler, and today let's look at the claim that mutations in shared pseudogenes in chimps and humans support the idea that they descended from a common ancestor. Pseudogenes are non-coding DNA sequences which look like certain genes, but with differences believed to be mutations. The idea is pseudogenes resulted from protein coding which lost their original functions or were
like how, I don't know, how she phrased it as believed to be mutations. Like, what would they be? Like, of course they think it's like diversity design from the get-go or something. Like, how would you test that? How would, what else could they be besides mutation?
Yeah, if it's not like the parents, like if you don't get it from one of the parents in that sequence, then it's a mutation, right?
Yeah.
Or some sort of recombination issue or something.
Yeah, it's DNA differences. That's fundamentally what it is.
Yeah, it's a mutation.
Yeah.
Okay. The interesting thing about this is...
Okay, you're very low in your volume speed.
Yeah, you are low.
Yes.
Yeah.
You need to shape up.
Are you kidding?
I'm not kidding. Patricia is much louder than you.
How about now?
You are much. That's better. That's better.
Yeah, but I'm just wondering why. Well, let me put the mic closer. I'll drag that thing over here. It's going to fall over. Okay. How about now?
Now you sound good.
Yeah, you sound fine. Yeah.
No, I'm all paranoid. Now you just threw me off my fucking...
Hey, I'll let you know. I'll let you know.
I'm just ending the stream now.
There for speed.
Oh, that's right. You are. Yeah. Oh, and you're there to tell me not to say certain things that I say to you sometimes when we're talking private.
Yeah, that we don't want to make sure it doesn't get over here.
Do you want to maybe back up to hear what she just said again?
Yeah.
I don't want to. She kind of talks fast.
descended from a common ancestor. Pseudogenes are non-coding DNA sequences which look like certain genes, but with differences believed to be mutations. The idea is pseudogenes resulted from protein-coding genes which lost their original functions or were accidentally copied somewhere along the evolutionary family tree, letting the extra copy accumulate mutations to become a pseudogene.
Okay,
so, if different kinds of organisms, say...
I do want to just quickly give her props for a decent explanation of what pseudogenes are and why they're used for evidence of evolution. So good job there.
Okay.
It's due.
Well, most you can give her credit for is reading it off a teleprompter, which it looks like she's doing. And,
True, yeah.
And then wondering if she wrote it. Maybe there's credits that'll say that she wrote the script. Which
maybe.
So...
Yeah. I'm just
congratulations to whoever put that script together for getting that part right.
Yeah, whoever did.
Yep. I want to point something out here, though. Thought to be mutations. In other words, she doesn't think they're mutations. She thinks there's by design. But they believe in genetic entropy, right? 100 mutations every generation.
So which is
which is it? Right. Which one do you want? Are there mutations or are there not? Now, I think what they claim is that the chimpanzee looks a little bit different than us, and they're going to claim that all of those mutations are required in order to make a chimpanzee look like himself and us look like ourselves, right?
Right.
That's one of the claims they have. Every mutation is God's design, but genetic entropy. Yeah, genetic entropy takes that and tosses it in the toilet.
Right.
Because if you accept genetic entropy, 100 mutations per generation, then you also have to accept that not all of those things are going to affect the phenotype.
Right.
Otherwise, we would be dead. We would have stepped off the ark and dropped dead.
But also kind of reminds one video Dr. Dan did about how if you believe created heterozygosity, you can't also believe in genetic entropy and vice versa, because they're
Right.
contradictory.
Yep.
So kind of similar to that. Yeah.
Okay. Now this person seems like
hey,
a
BS
really...
Lewis. Thanks for joining us today. Good to see you.
Oh, Nerdy and BS Lewis.
Yep. And Nerdy too. Hey.
Nerdy. Okay. Play more.
Chimps and humans have similar pseudogenes with the same mutations in the same spots. That's considered great evidence that chimps and humans share a common ancestor who had those mutations as well. Because statistically speaking, you wouldn't expect identical random mistakes to happen twice.
Yep.
So, do pseudogenes demonstrate evolution? Let's think about it. Critical thinking check number 1 is check scripture. And Genesis explains that God created everything.
I just swallowed my tongue. What?
That's kind of the same reaction. I like cooking skills.
Check scripture first. Okay. I don't know.
I always gotta
Very
check
good.
scripture.
So we would expect God to design completely useless genes, much less put them in multiple organisms. Still, we know creation didn't stay very good because of sin, so we wouldn't be surprised to see corruptions of originally good genes through mutations. Biblically, then, we'd expect pseudogenes either serve some function or lost their original functions, but we wouldn't interpret them as evolution.
Isn't that kind of what we're saying? They lost their original function?
Right.
I mean,
Isn't she kind of
agreeing
the
with us
point
there?
a little bit? Yeah.
Okay. It's interesting.
Evolutionary leftovers, because that would contradict a clear teaching of Scripture, which is check number 2, check
I
the
also
challenge.
want to point
Jumping
out
down to check number
that she just promotes pseudogenes are a result of the fall, basically, and genomic decay and whatnot.
Right.
I mean, that would explain the pseudo of it, sure, but it wouldn't explain the shared mutation aspect of it, which they're going to go over their thoughts on that specifically in a little bit, I think, but just want to point that out that so far they haven't addressed the actual problem yet.
Right.
But anyways,
Yeah,
a minute and a
coming
half
up.
in, so yeah.
Check for propaganda. Who is delivering the message?
Ironic.
Is it AIG? Okay, if it's AIG, then it's propaganda. So I'm passing that check.
Five.
What BS?
Propaganda.
Wait a
Can
minute.
you highlight BS's comment?
What were the other
Yeah,
three? Okay, she did 1. What happened to 2, 3, and 4?
Say that they applied for different videos.
Okay.
It's kind of
You
the
ready?
thing they do in these videos.
Oh, so this is something they do all the time. Okay.
Well, in this series in particular.
Yeah, check for propaganda. You go up here and you look at the 5, and you go down and you go down 3 lines and you see answers in Genesis, and you can be pretty sure it's propaganda. So she's right on right there. So I agree with her. Okay.
Can you?
We can apply the question, is this
I like what?
Oh, sorry. Just highlight BS's comment real quick. That's basically what I was saying. When your first critical thinking is check scripture, I don't think you went thinking. Yep.
Yeah, I don't think so. And Bree is here.
Hey, Bree.
Sure knocked him dead last night. 400 views.
Oh, that's
50,
right. She
60.
did a review on the tour debate too. And
Professor Dave stopped in, and Aaron Ross stopped in.
Oh wow!
I mean, it was pretty incredible. Yeah.
Darn, I'm sad I missed it.
Yeah, and Bree called me a half an hour before just to rub it in. Nah, I got Aaron Ron, Professor Dave, and you don't. I know what you were doing, Bree. Anyway.
Can you summarize like what they talked about, or is there too much to cover?
I don't know. I was too green with envy to actually listen to anything anybody said.
Oh, okay. We'll have to review it again some other time.
Yeah. No, it's a good video. Go watch it.
Okay.
They got into a whole like an hour of pre-show stuff with Professor Dave and Art Raw, and then started reviewing the video with Jay Bundy. And
how long was the whole thing?
3 hours. 3 hours.
Oh, okay.
It was a long one. So anyway.
Where'd Scott
go?
go?
Is he gone? Oh my God, his computer must have crashed.
He just left.
Oh, maybe,
Probably.
maybe angry.
We'll have on without him for now, I guess.
I don't know how we're gonna do it. Who's gonna keep me from saying those things I shouldn't say?
I guess I will.
Yeah, step up to the plate, Andrew. Okay, gonna proceed?
Sure.
This is a good one.
Message true or false, because to ask, is it true that pseudogenes have no function because we don't know what their function is? And like we saw with other types of so-called junk DNA, the answer is no. Saying pseudogenes are useless because we don't understand their use would be an appeal to ignorance fallacy.
Okay, pause.
On to check 6.
Okay, we gotta talk about
it's unfortunate then that people who use pseudogenes as emma descent don't use that line of reasoning. Yes, it would kind of sort of be a fallacy. But I guess that's a whole other thing about junk DNA altogether. But the reason we know that they probably don't have any encoding function or any functional related is usually, well, sometimes, but they often don't have their promoter there, so they can't be transcribed at all, basically, if they're missing the regulatory for them to be expressed to begin with. And second, they also have a lot of frame shift and premature stop codons caused by mutations because, if you mess up the sequence order there just a little bit, then you're probably not going to get a functional protein. So kind of a double whammy there for a lot of pseudogenes. And that's the big reason why we're pretty sure they don't have function. Not, we don't know what their function is yet, so we're just going to assume they don't have any function. And of course, for a lot of them, we know they don't have function because we tested them individually.
And to add on, DDX11L2, the famous chromosome fusion pseudogene in the very latest genome that they published on NCBI, you run into stop codes like 3 or 4 times. You just get started, stop, like 17 amino acids, stop code, another 20, stop code. It's just that no matter where you start, no matter where you find the start code, and you can start at different start codes, right? But it's going to start at the first one, and it runs into stop codes. So it's filthy with stop codes, but if you go back and you compare an older genome, they don't have those for some reason, like 37 or something like that. But I'm putting a lot of that together. That is definitely a pseudogene. Yeah. The other reason it's a pseudogene is there are massive numbers of variations between individuals in the human population. The other reason it's a pseudogene is because it only represents about 1/15 or 1/12 of the entire length of the actual DDX11 gene. I mean, it's the area that it represents is in the what do they call it? The end of the gene that gets all messy.
The subtelomere region.
Well, no, that's where they find it in the genome, but like the disordered region of the gene, the ones that doesn't fold into nice alpha helices and beta sheets and doesn't have active domains. So it's all in that basically that little junk piece that kind of sticks out on every protein. It just kind of wags around. Doesn't really seem to do all that much. So anyway, so that we have really good evidence that is a pseudogene. Really good evidence that it's a pseudogene.
Oh yeah.
So, and we got to check them all, and of course we are checking them all. The first one you mentioned can't be transcribed or doesn't have a promoter. That would rule out about 1/4 of them. Yeah, and we got 3/4, and out of those, we're going to find a lot of them that are functional. I have no doubt.
Yeah, that
But-
doesn't mean that all of them, therefore, are functional, though.
Right.
Or,
we just
or
have to wonder
near the
function.
number that they would need for creationists to this one.
Yeah, but they have that non-synonymous to synonymous ratio greater than 1, which also indicates that there's a problem.
Yeah, that too. Yeah.
And you don't just find that between us and chimpanzees. So they can't. You find that between me and you, between individuals in our population of humans,
Right.
and it's also where you can get a value for that. So
Yeah.
that kind of blows their idea out of the water that God did it
because
that
there
way.
are a lot of mutations at the rate expected they would be if they are neutral.
Right. Yeah, I think they like to imagine that chimpanzees have big lips because of some of those pseudogenes, right? that created
Right.
some whatever. But they're going to have to show us how that works because, yeah.
Hey, Speed, we have a question about your choice of thumbnail. Why is your thumbnail of a dude's junk?
Well, it's not really a dude's junk unless it's just that if you look at the psychedelic picture, it kind of like emphasizes his junk. And I did see that after I used the thumbnail. But what I was looking for there is I typed in fake genes, Geans, and that's what I got. So that looked like genes that weren't really genes. And I'm sorry.
Because
Then I thought, okay, it looks like that guy's junk is sticking out. And I thought, I don't care. Right? I'm going to let that slide because, I don't get all weird about other guys' junk.
But it amused BS at least, so we got one there.
It's a pseudogene, man.
Would it be safe to say
yeah,
that maybe
so
some of the wrong non-protein consumables got loose in your salad or something?
Something like that.
When you chose that? Okay.
Yeah. Well, looking at junk is kind of what we're looking at here. We're looking at AIG's junk.
Yeah.
Yes. Yeah. You can put it that way. You could.
Yeah. Okay. I did like a metaphor.
And we're barely halfway through.
We're barely halfway through. We're 1/3 of the way through. A little.
Oh,
over.
yeah. 1/3.
Interpretations. What's the observational science about pseudogenes? What facts can we observe right now? The fact is that different organisms have some similar non-coding DNA sequences, which themselves look similar to certain coding DNA sequences. The interpretation from historical sciences is that these non-coding sequences are largely useless evolutionary leftovers. So, what are some assumptions behind this explanation? Besides assuming that one kind of creature can evolve into another and that evolution can produce genes in the first place, it's assuming that pseudogenes are non-functional.
I don't know about y'all, but I don't think like testing pseudogenes and seeing that they're there and all the business behind that like assumes evolution or the kind. Is it just me?
Yeah,
It seems
is
like
pseudogene a topic for evolutionary biologists or for geneticists?
It can be both, but like searching for pseudogenes and how they're shared between species, like assuming that the kinds of organisms evolved and that new gene, like what that's another thing. Like, what do new genes have to do with pseudogenes? Like, sure, they can involve the same duplication process going on there. It's just an off-topic point there. I don't know why. I don't understand that point.
Well, she says the interpretation from historical science. What the hell is historical science? These
the
non-coding
they don't like.
Yeah, are largely useless evolutionary leftovers. No, I don't think anybody's assuming that because a lot of them are what you would call what these guys in this paper call pseudogenes, P-O-T-O genes. Have you heard that term before? I just run into it today.
I've heard of potted meat, but not potted genes.
Pseudogenes, potential genes.
Oh,
yeah.
And I don't hear gene language very
yeah,
well.
and gene duplications can if one of them mutates, the other one can step up and do the job. So sometimes that happens.
Yeah, so it is somewhat related, but it's not like we're not assuming
Uselessness.
And we can't observe processes, so we're not assuming that either. I don't know.
Yep. You know, the ultimate test is to not knock them off.
Yeah, that too.
You know, and we've done that in the mouse, and I think we've done it. We might have done it in Drosophila. Knocked a lot of them out and had no effect that we can tell on the organism. So, but Standing for Truth would say, "Oh, but you haven't your housefly or fruit fly has not flown to the Arctic Circle. And if he flew to the Arctic Circle, then one of those pseudogenes would step up and unthaw him, right?" Do you know about that argument?
Said that.
Well, he said that onions, right? The reason that onions have a 50 billion nucleotide genome and garlic has like a 12 is because the onion needs all those genes in case the onion happens to be growing at the Arctic Circle or, you know, way up north. And then all of those pseudogenes
works.
I know. Yeah.
I mean, all those Inuits have big onion patches out by their igloo.
Right.
Okay. I didn't know that, but that explains it.
I know. I get where he's coming from with that argument, because he thinks, there needs to be some redundancy there for designer purposes, but like biology wise, that literally no sense.
Yeah.
I just can't see people like that having read anything from their biology books in college. And I just wondered where that BSc that she's carrying around actually came from.
It's 16 billion base pairs for the onion. And garlic is closely related to onions. In fact, it would be the same created kind, I think, in their book, right?
Yeah.
Oh no, it has a large size too, so that's also 16.9. But
one of the members, onion test.
Yeah, one of those members of that family have less or a much smaller genome. That's allium, I think. Anyway,
won't the garlic need more genes to help it ward off vampires?
Yeah, it's got to have extra genes.
You would think.
Yeah. High heterozygosity, 1.69%.
I picture a little dial on their computers that measure the heterozygosity of things.
91.3% is repeated sequences. So there you go. Anyway, okay.
Getting back to what's real science and what's just made up words by propaganda people. Heterozygosity, is that a genetics word or is that a made up word?
No, it's a real genetics word.
Okay.
God damn. These
real term.
You know, do I have to start banning people out in chat because of this? I mean, I look at that and I see a pair of psychedelic shorts. I mean, I'm not looking at the guy. Come on, guy. Get your minds out of the gutter. God damn it. I can't win. Okay. Moving on.
Explanation. Besides assuming that one kind of creature can evolve into another and that evolution can produce genes in the first place, it's assuming that pseudogenes are non-functional and that the same mistakes wouldn't happen in the same place twice. To see if these assumptions are valid, we have to dig deeper into the observational science. You can do that in much more detail through the resources linked to this video. But to give you an idea, the more researchers discover, the clearer it's becoming that many so-called pseudogenes are functional. In fact, part of the reason why they might look like coding genes may be that RNA transcribed from them helps to regulate those coding genes. Recent ENCODE data lists pseudogenes as a category of functional RNA binding sequences. Some pseudogenes might even be coding genes expressed only in certain tissues under certain conditions.
Okay, this is an example of them using what evolutionary biologists have discovered to say that evolution didn't happen, right?
Right.
So I mean, they discovered that some of these pseudogenes create an RNA which ends up being a microRNA that attaches to one of the mRNAs and shuts it off. So some of them have a regulatory effect. And depending on the ratio of which ones you have in the cell, you're going to end up with different cell types. But that's exactly what we would expect from evolution, basically.
Right, and you can tell that they have regulatory functions because they're under purifying selection. Their sequence isn't going to change all too much compared to other pseudogenes where they're mutating like crazy. So it's kind of comparing apples to oranges here. Like they're not exactly equivalent. You can't say
like
that one does this, so therefore all of them maybe do this. Like,
scientists have now discovered that DNA and genes are responsible for how organisms differ. Therefore, evolution can't be true. So I mean, they say something that we know and that we're perfectly aware of and have discovered over the last 30 years, and it's just like, come on.
And of course, you get that little encode mentioned in there since we're talking about junk DNA.
Yep, a lot of it transcribes. And that's because the promoters aren't that complicated. The TATA box and a few other things that allow a gene to promote, basically get started transcribing, is pretty common. We have a lot of junk proteins floating around in our cells.
Right. That would only apply for the ones that can be transcribed, which I said earlier, a lot of them can't. So it doesn't apply to all of them again.
Yeah, and that junk is actually junk drawer junk. Yeah. It's not useless. It is how evolution happens. The DDX11 gene, which you find in the subtelomeric regions of all primates, there's a large family of those genes that have duplicated, created a whole host of pseudogenes besides that DDX11L2. And there's like a whole family of genes, and they can and actually they can look at that and see phylogeny of primates in those genes. And I have a paper on that. So anyway,
make their functions harder to detect. Now, some researchers apply evolutionary assumptions to say harder to expressed only in certain tissues under certain conditions, make their functions harder to detect.
Okay, so what? We check all tissue types.
Wait. I mean, you don't need to, beyond that, you don't need to know that, okay, it's expressed or whatever to know that it's functional. Like you can just look at the genetic sequence itself. Like, you don't have to always look at whatever it's doing in a particular tissue or all the tissues versus, I don't know. That just doesn't make sense.
Yeah, but it helps to find out where the heterochromatin is, where, basically where cells differ, right? So we look at all 200 tissue types.
Right, yeah, it helps, but I'm saying, like, it's not by itself the key thing here. Like, it's not the only way we can do this.
No. Yeah, she's saying, they're hard to detect, so evolutionists assumed that they weren't functional. It's like, no. Unless you've looked at every cell type, you're not going to make any assumption at all.
And again, they're confusing assumption with inference here. Like, okay, this is what we can detect so far, or what we figured out so far. So we're going to infer this as the best explanation for so far. That's not the same thing as an assumption. Like, they always make that conflation.
Why is that YouTube video so? What am I looking at here? Wow. Why is the URL so big? The URL on this is really huge. I don't know what
it is.
Yeah, really odd. Really odd. Anyway, okay. I was going to put this in our description.
Sometimes Mike makes these references to things that he sees, but nobody else does. And that's what I was wondering about that URL is, are you talking about this video or some other thing that we don't have access to?
I always ask that you guys try to keep up, try to keep up.
By the way, I'm gonna have to share my screen.
Uh-oh.
Hold on for just a moment. I got to find where's the screen share?
Oh my God, that guy's junk is highly visible. I just looked at my thumbnail in a smaller scale.
Okay, there. This apparently is an example of gene duplication.
Yeah, exactly. That's gene duplication.
Well,
Yeah. Thanks to Bree from Hive
thanks,
Sci.
Bree.
He is very scientific. He knows all about gene duplication
He?
and pseudogenes.
Don't you mean she?
Gene Simmons.
Yeah. Oh, you don't know who Gene Simmons is? That's so sweet.
That is sweet.
There's the pseudogene.
Yeah, there's a pseudogene.
Oh.
All right.
My son and my daughter-in-law were hanging out with him out in Las Vegas not too long ago. And she said he's kind of a pig, I guess.
Looks like a balance.
Yeah, I never thought he was particularly likable.
Yeah. He's from the band Kiss, Andrew.
Oh, okay.
Yeah, the guys that paint their face like that. And yeah. And
Oh, I
make
thought you
music.
guys were talking about Bree, or that's what I was talking about.
No, pseudogenes.
Oh,
Pseudogene Simmons. Yeah.
okay.
They do.
Yeah,
I think
yeah.
I'm lost. Okay.
Yeah.
Oh,
But anyway,
here you go. Here you go. B.S. Lewis. Spot on. And we caught J. Patrick.
You mean a homophobe detector?
Yeah, homophobe detector too. Jeez.
We only endorse and laugh at homophobe detectors, and that's what we took him to mean on there.
Love you, Jay. I want to kill you, but love you.
I don't know why you want to kill Jay. He's pretty fun guy.
Yeah, but yeah.
Just because you disagree about philosophical issues, you want to kill him?
Yeah.
Okay,
Hey,
well,
I've got a joke for you.
it looks like everybody is tweeting their RIPs for Tina Turner.
Oh,
Do you know who Tina Turner was, Andrew?
Nope.
What's love got to do? Okay, I'm not going to try to imitate anyway.
Oh, okay.
The song "What's Love Got to Do with It"? Do you remember that one?
Yeah.
She's the one who sang that.
Oh,
yeah.
okay.
That's not her most famous song. She was around for decades.
And she was very outspoken about domestic abuse. She was a victim, and she became very famous for part of that. Plus, there's singing and dancing, and was just died at 88 years old.
And of course, she was an icon for her role in, I forget the name of the character she played in Thunderdome.
Oh,
Mad Max Beyond Thunderdome.
Aunt Chaos or something like that.
Yeah, or something like that.
Exile, or I don't know, Anti-Exile.
But yeah, she played a great role there.
Yeah.
Yeah, she's a icon of
Yeah, it was of an
old folks generation. Yeah,
big loss. Big loss. Okay, we're gonna go on.
Okay.
Now, some researchers apply evolutionary assumptions to say that pseudogenes must have evolved such functions after the fact, although a study which looked for evidence of this didn't find much.
Okay, what is she talking about?
I don't know.
Okay, you're suddenly quiet again.
I am?
Well, you are quiet.
Oh,
Yes.
facing
Yeah.
away. How do I get quiet
And-
again?
Okay, let me just.
Hang on a second. Let me shut off Chris. Oh, let's see here. Start recording. Chris microphone. Oh, it's already shut off. I don't know.
Well, you just got louder again.
What the fuck? Okay, maybe it's canceling me out.
You've been canceled.
I've been canceled. Okay.
No, but even YouTube doesn't like the shorts avatar.
What evidence is she
Thumbnail.
talking about that showed that they didn't find much
I don't
evidence
know.
that
She doesn't, like, they had the papers, yeah, right there, but they don't give a proper citation for them.
Yes.
Okay,
Like,
we get
On a scale of
You're
1 to
talking about-
5, on a scale of 1 to 5, everybody in the super sub chat are not, that thing that goes up and down the side chat. Everybody in the side chat, do a rating between 1 and 5, 1 being the least, 5 being the most confident. How confident are you of this AIG person's analytical skills?
Yeah.
What's your confidence level? 1 for very weak, 5 for very strong.
Like you were saying,
do you think that they can do
I doubt it was her who made that, and it was probably someone else, and she's just...
Well, that's true. Back to the script writer.
Yeah.
Well, she lists all of her digging deeper sources: Tompkins, AIG.
Yeah, those ones are all AIG sources, but they're actual papers below them. Yeah, right there.
Yeah.
And has anybody seen Erica's critiques of Tompkins? Is it Tompkins?
Yep.
Yeah, recently on the Gutsick Gibbon channel.
Yeah, she's gonna do an upcoming video about that one.
Yeah, she really nails him to the wall.
Yep.
So Tompkins is definitely not a great source for anybody who wants to relate scientific data. It's not trustworthy. I mean, he might be right, but he's been wrong and boldly wrong on so many things that
Right.
you definitely have to double check him with other people, and other people are saying he's wrong.
Yeah.
Yeah, and if you look at the ads,
But you to whatever she just quoted, that it didn't what she's saying it said.
so in the abstract of one of the papers that she cites by Belakurov and Ayala, Francisco Ayala, at the end of the abstract, they say, we agree with the proposal that pseudogenes must be considered as pseudogenes. That's where I got the word from. I.e.,
Potential
DNA sequences with a potentiality for becoming true genes. That's one of the papers they cite, which do not agree with what she's saying. That's just the first paper. And I'm guessing that none of the other papers that I look at are going to agree with this. We looked for evidence of this and didn't find much. Right.
Well, like let me give an analogy. Let me give an analogy. I don't use analogies much, but I think this is a good one. It's similar to one they use on Biologos website where they talk about pseudo. Like, okay, suppose you're in a house and you can figure out, okay, this house was after you or before you bought it. Basically, like the previous owner made some changes here and there. Let's say the back room, the plumbing to the not is the same wiring and whatnot, but everything else around the toilet and the what was once a bathroom has been remodeled into a room. Now a rational person would say, okay, it has a new function. It's a living room now. It still has the signature, the remnants of what was probably a bathroom before. So in the same way, analogous to pseudogenes, you can say, okay, they may have a new function now, but you can still tell that they were previous a different kind of protein coding gene that lost their function, or their original function, and now have gained a new function. And so it doesn't really depend on the functional aspect of it. You can tell either way that the history is still the same either way. So I hope that makes sense.
Yeah, like you go into the
the
dining room. You go into the dining room and you find that one of the chairs underneath the table is a toilet with a plywood on top of it.
Yeah, exactly. Yeah.
Yeah. Which would be a little weird, but
yeah.
I can
now,
see.
Of course, organisms or have appliances in them or machines or whatever, but
they're not machines either, damn it.
Yeah, but I think that hopefully that analogy gets the point across. Yeah.
Okay.
Although a study which looked for evidence of this didn't find much. The researchers concluded that because they didn't see much evidence for more functional motifs evolving in pseudogenes, pseudogenes must not have functions. But again, that's an interpretation based on evolutionary assumptions.
Why?
How is that evolutionary assumption?
Okay, if you don't see functional motifs, then you could assume that it probably doesn't have function, right? If you don't find any functional motifs at all, or any functional folds, or functional beta sheets or alpha helices, you don't find anything like that. It's just a disordered gene or whatever, or it doesn't seem to have any motifs that we've ever heard of. We can not assume, but we can infer that it may not have function, right? We can't assume that because we may find some functional motif that we didn't know about before, right?
Yeah.
But we've cataloged like 50,000 of them or 100,000 of them so far.
I would need to see the textual trail of a scientist assuming, based on their evolutionary bias, that something doesn't have a function, because there's also, for the same thing, there's also evidence that they didn't see a function and therefore concluded it's not functional. And that may not be, it may be one of those black swan kind of fallacies that you're engaging in, but it's not based on evolutionary assumption. It's just based on thinking that because it doesn't have a function that I can recognize now, that it won't have a function that I can recognize later when I have better information. So it's not something, it's not necessarily based on evolutionary assumptions. And if you're going to assume somebody's state of mind, if you're going to presume upon that, then document why you think that's true.
Yeah. Extraordinary claims require extraordinary evidence, after all.
She's implying that we just looked at them and we didn't find any functional domains, so we just discarded them. We'll never look
up our hands and non-functional.
There's a number of databases: SMART, Uniprot, PFAM, ProSite, and the other one is what the hell's the other one? There's one more. Oh God, I forget what that is. Anyways, a number of sites where we've documented hundreds of thousands of functional domains, and we can pick them out. You can blast and search for them, and you do a pretty good job of analyzing any new gene that you find. So
yeah, we know how to do this. We're not clueless about how to find function in a genome.
We're not going to just assume it doesn't have function because evolution.
Yeah.
Okay.
Motifs evolving in pseudogenes. Pseudogenes must not have functions. But again, that's an interpretation based on evolutionary assumptions. The observations remain that pseudogenes play numerous roles, as multiple other studies document.
But
now, what about this?
All of them have those roles.
That's a lie, because if they find a function for a pseudogene, it comes out of the pseudogene database. It's no longer considered a pseudogene if they find a function. But she's implying that, what the hell did she say exactly? The observations remain that pseudogenes play numerous roles. Okay, some of them we have found roles for, like a handful. I think when I searched one time, I think I found like 12 of them that we found function for.
Out of 1,000.
And so we're chipping away at that 12,000 pseudogenes very slowly if we found 12 out of 12,000. So I'm pretty sure it's in the hundreds now, but I mean,
same idea.
That's just a lie. He's a liar.
Roles, as multiple other studies document. Now, what about the assumption that the same mutation wouldn't happen twice in the same place? Actually, as you can learn in more detail elsewhere, particular regions of DNA sequences seem to be hotspots for certain types of mutations. So, assuming these differences between genes and pseudogenes are mutations, it wouldn't necessarily be unreasonable for them to appear in multiple organisms. For instance, the fact that humans...
Okay, that would be extremely statistically unlikely.
Right. Well, notice the wording she used. Hotspots for certain types of mutations. She didn't just say they're spots for mutations in general, or that mutations will occur more often there. She said for certain types of certain mutations. Like, no, they're not hotspots for certain mutations. Mutations stochastic or probabilistically either way. Like, just it being a hotspot isn't going to make more one type of mutation more likely to occur than other. This is the same problem with Donnie's ERV argument. So,
but if you look at if you look at these repeated sequences, right, repeated areas that are GC rich or AT rich or whatever, they may have a likelihood to one type of mutation, because if you have a lot of GC, what are you going to end up you're going to end up with something if you mutate that. And those hotspots, a lot of those hotspot regions are in those areas with vast numbers of repeats. And that's why they're hotspots, because they have these repeated sequence. It becomes very difficult to repair a piece of DNA like that. And it became,
sure, but the genes aren't repeat sequences, though, so it's not exactly
pseudogenes. Right. That's the other thing. Pseudogenes are like they're in that 2% of the genome that ends up being a gene.
Right. They're gene
differences between genes.
Yeah, lots of little lies going on here. In order to do biblical thinking, well, you got to jump through hoops.
Genes and pseudogenes are mutations. It wouldn't necessarily be unreasonable for them to appear in multiple organisms. For instance, the fact that humans and apes have similar mutations in GULO pseudogenes supposedly means they share a common ancestor. But humans and guinea pigs also share 36% of the same GULO mutations, showing that similar mutations do appear in different lineages. Besides,
so I went down to because I thought, okay, that sounds a little iffy, because those mutations occurred the same in primates and guinea pigs. That would seem a little weird. So if you don't mind, I'd like to share my screen. After looking into it and looking over some reviews of this particular claim, it seems to be from some maybe one or two papers in the early 2000s, around 2003, I think. And what the researchers of that paper did, let me just scroll down here.
I'm going to hit you up for the name of that paper, Andrew. Name and author. That's all I need.
I think it's INAI 2003 is the one. INAI 2003. I also have one Pandas Film article here where they talk about it too, if you want that. Okay. I can send you these links afterwards.
Yeah, okay. We got to put them in the description.
All right.
You talk. You can talk now.
Yeah, so what the researchers did was, or the researchers of that paper did, was they compared the rat GULO pseudogene sequence to the guinea pig sequence and the human. And of course, they found, the guinea pig and GULO pseudogenes, the sequences had the same mutations compared to the rat, or they were more similar compared to the rat sequence. So they therefore concluded, maybe these mutations didn't occur independently, or they're, like what she was saying, basically. However, and this was actually pointed out to the researchers afterwards, and they retracted their claim there. I don't know if they actually did anything else with the original paper, but that's neither here nor there. Basically, what they're doing and what the other researchers showed is that if you compare those same mutations, but now across a larger sample of mammals, so it's not just the rat, the guinea pig, and the human. Now you're comparing it to other mammals like cows, pigs, and a mouse against the rat, for example. You find that those mutations represented in the spots by these red air that occurred in the human and that the human and guinea pig sequence share, like AA right here, the ancestral sequence for nearly all all of these when you compare it to like the cow or the pig, for example. And it turns out it's actually the rat sequence that is the more derived sequence, so to speak. It got the different mutation. Everything else just inherited the ancestral version at that spot. So it's because the rat sequence is actually evolving really fast compared to some other mammal.
Yep.
So it's the rat and the mouse sequence that are the outliers here, not that the human and pig are more similar to one another. They actually carry the ancestral version of the nucleotide at almost all these spots that they claim were more similar compared to the rat sequence. So I think that's where that 36% number comes from, and that's why it's not exactly accurate. So no, in either case, the same mutation, the GULO pseudogene, independently occurred independently in guinea pig primates. Like they're different mutations altogether. And after this video is done, I'll go more into that. I just want to note that now, because that claim she cited there, and this was known, like I said, back in the early 2000s. So I don't know why they're still citing this claim in 2020 or whenever this video was published.
Are rat and mouse the same kind, according to the Biblicans?
I would assume so.
Okay, rats have 42 chromosomes and mice have 40. Aha. Isn't that similar to chimps and humans? We have two less. Okay, we have two less chromosomes than the chimp, and mice have two less chromosomes than rats, the same created kind, rats and mice. And not only that, but I think they did a study in Germany, and they found across populations of mice, they found all kinds of variation in the number of chromosomes that they have. It isn't just 40 and 42. It's typically
so a lot
40.
of variation.
But okay, so there was a chromosome fusion in their created kind. They're one created kind. One of them had 40 and the other one has 42. Oh no, God decided the rats need 2 more chromosomes because they're bigger than mice, right?
Right.
But yeah, he needed to do some more architecture, so he just added another chromosome.
So I just posted the Pandas Film article where I got that little picture from in the private chat, if you want, in the description too. Among the other things I'll show in a little bit.
Oh, that was on Pandas Thumb. Okay.
Yeah, Pandas Film. Yeah.
Got it.
Where they were talking about this specific claim. That's where I went down. So.
The revenge of the guinea pigs.
Yeah.
Okay. Let me get that done, and then we'll play
cow, rat, pig.
Man, bear, pig. I like man, bear, pig. You watch South Park, Andrew?
No.
You don't get out much.
I watch some stuff. I just don't watch that kind of stuff. It's just not funny to me.
Oh, really? South Park is really good political commentary and very balanced on top of it.
That's probably another about politics either. So.
Yeah, well, you better care in 2024, damn it, or we're going to end up with a problem.
Yeah, you need to care, because we basically, my generation just screwed up the whole world for you.
Yeah.
And probably not going to live long enough to help do anything about it. So, oops,
well,
sorry.
Not mine. I'll try.
Well, yeah, but I'll be dead. I don't have to face the consequences.
Okay, Big Bad Mama has another review of the tour debate.
Oh, wow. Lots of people reviewing the tour debate.
Yep. Yep. It's going to be a thing for a long time, I'm sure.
It breathed new life into the skeptical community.
Yep. Yeah, it got us all going. And it is my opinion that everybody better fucking quit picking on Professor Dave, or I'm going to get really pissed.
Okay.
Because I thought he did exactly what he needed to do. He needed to call that charlatan out. Okay, go on, play the rest of it.
I think it should be almost over.
In many cases, the fact that organisms display similar pseudogenes hints that these sequences do have functions. Ultimately, a biblical interpretation that pseudogenes represent useful designs or information losses makes sense of the observational science and requires no evolutionary explanation. For more on how to think critically
Okay,
about
pause
challenging
the
messages,
button,
you
damn
can
it.
access my other CT scan videos packed with
The use of the word biblical
Yeah.
is so laughable, because she's saying that the Bible has a position on pseudogenes.
Right. A biblical interpretation that pseudogenes represent useful designs. Show me the passage.
They didn't even know you should wash your hands after you sneeze in them.
Right.
Right. What did you notice that other claim she made too, Speech? She said because they're shared, they're probably functional. Like, does that make any sense?
No, that doesn't.
Like, that doesn't follow. Yeah, that struck me as odd too.
Yeah. So what else does she have to say? Is this all just that helped me as a Christian
That's
student?
promotional stuff.
Yeah, let's listen to the part. Well, I want to listen to that part.
For more on how to think critically about faith-challenging messages, you can access my other CT scan videos packed with tactics, tips, and tools that helped me as a Christian student at Secular University. Thank you for watching.
It helped you ignore everything your professors were trying to tell you.
Yeah.
I mean, if you want help ignoring everything that they're trying to tell you, check these videos out. I mean, these are great.
Yeah.
They got all these little snotty kids coming to college going, hey, I don't have to listen to him. I just have to pretend I'm listening. And so they'll get their grade and go on.
Ooh.
I hope they had to pay full price for their college.
I hope so.
If you like this video, hit the like button. But if you don't like it, of course, I just gave them more ammo, didn't I? Oh my God, this got 446 views. Yeah.
Do dislikes even count on YouTube anymore? I don't know if they do.
Yeah, they count. I think they count as they feed into their algorithm. It means you watched it and you commented on it. You felt strongly about it. They want you to feel strongly about things, and
that's why I don't like her. Or that's why I don't like, because I don't want to give them the attention.
Yeah, okay, I figured that out a long time ago. Yeah.
So I linked this BioLogos article. I linked this Revenge of the Guinea Pigs and Panda's Thumb. Let's see. I linked the AIG thing by what's his name? John Woodmore Rape.
I think that's the one where the 36% claim came from, right?
Yeah, I think so.
I think that's where I saw it.
Yeah, and he links this paper, 2003 paper. They link a lot of 2003 papers on pseudogenes.
Yeah.
But anyway, this is something that we can look into further, and we could drill down and find a few pseudogenes, like find a sample of say 12 of them, and go to town and figure out what we can figure out. I've done a lot of work on DDX11L2 and found it to be absolutely not a fucking gene. Absolutely no fucking way. In fact, I'm so certain of it that if somebody wants to pay, I will volunteer to have that gene knocked out in my entire body if you can figure out a way to do that. So, I mean, I'll take one for the team here and be the knockout test on DDX11L2. I don't know if you know it or not, but Standing for Truth has challenged people to do that. If you don't believe it, and like I'm like, okay, I'm all for it, Dan.
He sure does have some bizarre challenges.
Right, he does. I'm 72 years old. I got nothing to lose. Knock that fucker out. I don't care.
So before we do close out today, I'd like to say I can share my screen one more time.
Yes. Oh, you have
one more.
Yeah, go ahead.
Are you seeing it right now?
No, somebody has clicked the button. Scott's been like resting on his laurels, I guess. We both click at the same time, but Go ahead.
Can you see my mouse?
I can.
Okay. So I was going to talk about some other ones, but I'm going to save it for the gulonolactone oxidase pseudogene. That's the GULO pseudogene they were talking about. So pictures here just to show you real quick, and I'll link where I got them from too. So here.
Go ahead. Oh no, go ahead.
Oh. Copy this. No, I don't want to copy.
Sandrock black spot.
These two are both from the sandbox thing. I'll copy that in a sec. My internet's too slow. Okay. So notice. So human, chimp, macaque, and galgo. I don't know exactly what. I'll have to look at that. I think it's maybe a more basal primate, I guess. But here's one sequence, and you can see human, chimp, all share this deletion here of some of the, and you can also see that here compared to the rat sequence and the other primate sequences. They're missing exons 1 and 2. Gibbons are also missing some other exons, but those aren't shared with the other primates. All of them are also missing exon 5, 7, and 10, I believe. Yeah, I think those are the 5. So yeah, so those are the primates are missing. And just to compare it to the Guinea, hold on. Sorry. Let me use this scroller. Let's compare that to the guinea pig sequence, for example.
Was that an image of Tompkins?
Oh yeah, which one was looking earlier? Let's see. All right, so here is the human, chimp, macaque sequence and the rat sequence. So the rat's fully functional, obviously. It's not knocked out or anything. So yeah, human, chimpanzee, macaque missing exons 1, 2, 3, 5, 6, 8. Oh, that's a little different from the. Oh. Okay, maybe they numbered them differently. I don't know, but in any case, you can see missing almost half of the exons of the original GULO gene, and compared to guinea pigs that are not. This was probably a more recent king of the GULO gene, so to speak, because it's not nearly as mutated or all the not nearly as many exons are missing compared to the human chimpanzee macaque gene. Now, of course, both of them have stop codons and other disabling mutations as well. But of course, if you were to look at the actual genetic sequence, like I was saying earlier, you'll find those are independent mutations at different sites. So again, the whole mutational hotspot idea, even if it did make mutations deterministic, it still doesn't explain the big differences between how the primate gene is broken compared to how the guinea pig or a few other species, how that GULO gene is broken. So I just want to make note of that here, just to add on to what I was saying earlier.
Yep. Here's another person confused by my thumbnail.
James.
Nobody gets pseudogenes.
Yeah.
God, people.
Welcome to the club, Speed.
Yeah. Yeah, we could go further into the GULO gene. Something that I have resisted learning about for a long time.
Oh,
for some reason.
Why is that?
I don't know. Found it Well, I saw other squirrels to chase, basically. I'm
Oh,
chasing
okay.
a new squirrel now. I'm chasing a brand new squirrel. I found a book on category theory in biology that called Life Itself by Robert Rosen. I'm pursuing that squirrel right now. Oh, I have so many squirrels in so little time.
Well, at least your squirrel has some substance to it. My squirrel has been Mrs. Davis.
Oh yeah,
on Peacock
I'm gonna
streaming.
have to watch that. I've been watching For All Mankind, the series. It's about what would have happened if the Russians had gotten to the moon first, and basically they make Nixon look like the asshole that he was.
He was kind of an asshole.
Yeah, he was a good president though, but he was an asshole. I mean, he did a lot of really shady things along with his good presidency.
Well, didn't he start the war on drugs specifically to to suppress and oppress the
hippies and black people
community? Yeah,
and college protesters. Basically, he wanted to take them out in one fell swoop by starting the war on drugs.
I'd say he was a president, but I won't add good to it.
Yeah, well, he got us out of Vietnam.
Yeah,
and he had to, basically. Couldn't do it any other way, but he did get us out, and he did a few other things that were, reasonable. But
reasonable.
We had a lot of good presidents until 2016. In fact, all of them were good up until 2016. They all had their points. They on one side or the other, they took us one way or the other, pushing us this way and that way across that line in the middle. And then 2016 happened, and that was kind of the end of it. So, but hopefully he'll be in jail soon.
Yeah.
And
so the Sandwalk blog post and the other paper, I got those figures from that I just showed, Speed. If you want to put those in the description too.
Go ahead and put that in there.
Talk about some other pseudogenes, but I fear I'm probably boring my audience now.
Well,
I hope the video was interesting to review at least.
Yeah. Might not be a bad idea to pick a few of those and drill into it and just. I mean, they claim that we don't. Yeah. They claim that James Tour didn't show the chemistry or show the science. They keep saying we don't show.
You mean David Farina didn't? Dave Farina didn't.
Yeah, Farina. Yeah.
It really wasn't about that. It's are we clueless?
Yeah. For some reason.
Just because Tour said he had to meet those 5 criteria doesn't mean that he had to meet those 5 criteria. Farina's job wasn't to satisfy what's-his-face's criteria, but to simply demonstrate that we are not clueless.
Yeah, right.
And the worst thing that James Tour did in that entire thing is he put up those 5 things, forming polypeptides, right? Number 1. Number 2, I think, was DNA and RNA.
Yeah, polynucleotides.
Okay, those 2 things are not separate line items for which we're clueless. Those 2 things actually work together. In most current most of our current hypotheses about abiogenesis, RNA and protein creation work together. And it's not something where you have to show that a protein can form, that's 1,000 amino acids long, and you have to show that it can form randomly by polypeptides, by condensation of amino acids. No, that was the biggest lie that James Tour told, unless he is so goddamn clueless about biology that he actually thinks that that's how it happened. Is he that dumb? Is he that clueless?
He's definitely out of field. Yeah. He's out of his field. So you have to take everything he says with a grain of salt, even though he knows organic chemistry.
Yeah. So if you can't show how a polypeptide can form, and then you can't show how the RNA can form separately, then but we can. The problem is that we can. Just not the two it isn't a slam dunk for the two amino acids he was talking about. He picked the toughest ones, right? The ones that are most likely to have two activated groups.
Yeah.
You know,
he probably did that on purpose.
But those isomers are really, they could be biologically functional. The isomers, even if it formed wrong, those things could be biologically functional. He would have to show that they can't in an abiogenesis world, right, where life is getting started. In fact, I think that is an area of research that we need to spend more time on, is looking at these basically heterochiral amino acids, and we have to look at small branched peptides, and we have to look at other amino acids besides the 20 we got, because I think all of those things were functional in abiogenesis, and we're not going to know the whole story until we look at a whole lot of other things, right? And then we can piece it. And of course he says, oh no, it has to happen. You have to have these things, you have to have this protein develop, and then you have to have this DNA develop, and then you have to put them inside of a cell membrane, and they have to be alive. We have to create life. It's just all bullshit. Oh,
Yep.
You got me started on that. I wasn't going to start. Yeah.
We're going to have to go do another hour and a half.
Yeah. We got to go away. Okay, I put those in the description.
All right, thanks.
Okay. Oh, didn't you have a link to the Sandwalk in your thing?
Yeah, I put those in the private chat.
Oh, was it there? Did I see that? Where's Sandwalk? Oh, there it is. Oh God, you are fast.
I am fast.
I'll put that in the description too.
I try to meet your expectations speed.
Yep. And have we met your expectations today, Andrew?
Yeah. What else can we do to meet your expectations?
No, I think we did good. I I just hope I bored the audience right now. I probably could have explained a few things better, but I tried my best.
Well, perhaps the audience would like to ask Andrew some questions to make sure that you're clear on everything.
Yeah, and before that, I'd like to say to the audience, if you were bored by that, then go back and listen again and think about it a little bit, because
go to your room and think about it.
We're not here to bore you. We're here to inform you.
Yeah, that's what I try to do.
Right, but if you don't listen to us, if you're sitting there twiddling your thumbs.
When I confront new information and have to think about it, I get really sleepy.
Yeah, I know. I do that. Every time I pop in a video on category theory, I fall asleep at about the 20-minute mark, and then I have to go back and start again, and I get another 5 minutes, and bang, I'm out.
You should only watch 20-minute category theory movies.
Yeah, I know. Yeah. Yep. Are we done?
I have nothing to
anything else you wanted to talk about? I'm good.
No, we don't want you to have to take your name off the video again.
You still remember that?
I'm never going to forget that, Andrew.
Oh.
Yeah. That's going to be a thing between us forever. It's like,
well, thank you. Let's thank Dave very much for his kind comment.
I appreciate that.
Oh, thank you.
Not a compliment. Thank you, sir.
That's what I try to do.
Thank you. He's
the one person I like to educate, but I feel like if I did become a teacher, I'd be a poor teacher. Oh, maybe just me.
Okay, so there's more comments about my thumbnail that we have to deal with before we see what James wrote. Oh, what did James write? James learned evolution from South Park. He's confused by the thumbnail. Okay.
They have evolution in South Park?
Oh, yeah. They talk about it a couple times.
Do they talk about it in a negative or positive light?
They have people saying really stupid things, and they have other people saying really clever things. Everything they cover from one side of the spectrum to the other, and they do a really good job if you get their humor. And Their latest season though is crap. They went over to HBO, and I think the writers have retired, but they're still writing. So the latest season just isn't all that good. But all 24 seasons are just jam-packed.
24 seasons. My goodness.
Good political commentary. I love
24 seasons of what?
South Park.
South
25.
Park. Yeah. Sorry, I was looking at a tweet.
Yeah.
That somebody had. Apparently, Marjorie Taylor Greene was in some committee or doing something on the floor, maybe filling in for the speaker. Somehow, in this reality, she's filling in for the Speaker of the House, and she tells people to have some decorum, and the entire Democratic wing of the House just breaks down in laughter.
As they should.
Yeah, she's
that woman is unhinged. I mean, how the hell did she ever get elected? I don't. Oh my God, how did Trump get elected?
I don't know.
I'm so confused. What is happening?
Just one of those cool jokes that just went way too hard, way too far. A prank that had unintended consequences.
Yeah.
Okay, guys, we're gonna shoot her in the head.
Okay.
All right, everybody in the side chat, y'all have a great evening.
Yep. Thank you all. Goodbye.